Letter 2. I heard a comment regarding a Big Bang if we don't evolve as pertaining to personal relationships, and I decided to do a discussion on evolution now. I know some or all the things I will mention may have been brought forth by others previously, however, I would like to bring some of them together in a semi-comprehensive analysis of the theory of evolution to demonstrate the fact that the theory is rather lacking. In fact, it is greatly lacking. Perhaps you would share some of this analysis in some way so that others may come to their own conclusions regarding the fallacy of the theory of evolution. First, however, I would like to expand on my previous letter. I would just like to say that I know He is the light. I would like to say also that I am not proposing what technology our Father uses, only that it exists. Call it ability if you like. Some people are just so wrapped up in their belief system that they cannot cognize what you say before they attack it, thereby missing the entire point. Another important consideration is that the model I have adopted for creation requires a parent universe, which is connected to the child universe via a black hole. This connection disappears as the child universe expands, dissipating the black hole. However, one must remember that a super technology that can create a child universe should also be able to connect them via other black holes or perhaps maintain the connecting black hole. Another term used for a black hole is wormhole. A wormhole is a tunnel speculated to connect different space-times. It is my opinion that they connect our universe with our Father's and are used to bridge the dimensions. Thus, the bright light at the end of a tunnel experience is some food for thought. To begin to analyze the theory of evolution, one must first understand just what it is and how it came to be. The theory is not based on any science, rather, it is based upon observation. Although science is also rooted in observation, it has provable principles, whereas the theory of evolution does not. It is particularly important that people understand that evolution is just that, a theory, and nothing more. The theory was first put forth to explain the observation that, as one goes further into the history of the Earth, fewer and fewer species are found. In addition, the life forms found become less complex as one travels back in time. The observations are made by digging into the Earth's crust, thereby unearthing the remains of various creatures. There is no science directly involved, although there is science in associated disciplines. One must realize that there is a quite simple explanation for the above observations. The generally accepted model of evolution is the tree of evolution, a graphical organization of creatures into a vertical classification starting with the simplest organisms at the roots and ending with the most complex in the bough tops. There is only one attribute of this model that can be accepted, and that is, the arrangement from bottom to top also corresponds with the time axis, with the bottom, or roots, being the farthest back in time. This tree model is better replaced with a stratification model, as the supposed intermediates between the phylogenetic groups in the tree are represented as branches with no actual examples to insert. The theory supposes that one group evolves from another through random genetic mutations and natural selection. The same mechanism is proposed for the formation of various species within a particular group. This process requires that the mutants themselves mutate until a new group is formed. However, no examples of these mutants exist, therefore, they can only be represented as pathways in the model. In fact, these pathways do not exist, thus, there are many missing links, not just a missing link between modern man and primates.
Which single-cell organism did mammals evolve from?
All animals, including mammals, evolved from a single-celled organism known as the last common ancestor of animals, animal LCA, which lived over 600 million years ago. We can't know for certain what this ancestor looked like because it no longer exists, but scientists study the closest living single-celled relatives of animals to understand how the transition to multicellular life likely happened. Based on DNA comparisons, the closest living single-celled relatives of animals are a group of flagellated protists called choanoflagellates. These microorganisms, which can live as single cells or form colonies, provide the best insight into our unicellular origin.
What was the next organism after the ancestor in the mammalian path?
There was not one single organism that was the next in the mammalian path after the single-celled ancestor. The transition from unicellular to multicellular life, and then to mammals, happened over hundreds of millions of years and involved numerous intermediate forms, with many lineages branching off at different stages. While choanoflagellates are the closest living single-celled relatives of animals, they are not our direct ancestors. As for the first multicellular organisms, two candidates are commonly discussed. One is comb jellies, Ctenophores. Some genetic studies suggest comb jellies were the first to branch off from the animal family tree, around 600–700 million years ago. And the other are sponges, Porifera. The morphological similarity between their cells and choanoflagellates make them a candidate.
Give an example of an intermediate.
Examples of intermediates between earlier vertebrates and mammals are the synapsids, often referred to as mammal-like reptiles even though they were neither true mammals nor true reptiles. A more specific group within the synapsids that shows a high degree of intermediate features is the cynodonts. The lineage leading to modern humans follows a path from synapsids, through early mammals and primates, to hominins.
What is an intermediate between the synapsids and man?
A simple intermediate is difficult to define because the evolution was not a single line but a complex branching tree. However, one of the most critical intermediates between the last of the cynodont synapsids and modern humans was the early primate.
What was before the primate but after the synapsids?
The transition from the cynodont synapsids to primates was not a single step, but a gradual process that occurred over tens of millions of years and involved the appearance of the earliest mammals. The first true mammals, which evolved from advanced cynodonts in the late Triassic period, were the animals that existed after the synapsids but before the primates. Early examples of true mammals include small, burrowing creatures like Morganucodon.
But there must be evidence of an intermediate between them since the intermediates had to survive long enough to mutate.
You are correct that intermediates must have survived long enough to evolve, and the fossil record contains examples that document the transition between synapsids and archaic primates, as well as the later transition to true primates. The problem for researchers is that the earliest mammals and archaic primates were small, fragile creatures that did not fossilize as easily as larger animals.
What was between cynodonts and the Morganocodon?
There were several intermediate cynodonts between the earliest forms and the earliest mammals like Morganucodon. The period from the Triassic through the early Jurassic saw a gradual transition, not a single species moving toward a goal. Key transitional steps involved advanced cynodonts known as mammaliforms, which include groups like the Trithelodontids and the Tritylodontids. It led to Probainognathia, a group of advanced cynodonts that includes the ancestors of mammals.
What is an example of an intermediate cynodont?
The Probainognathian most commonly referenced in the context of mammal evolution is the genus Probainognathus. The name itself, meaning progressive jaw, reflects its importance as a transitional fossil with a combination of reptilian and incipient mammalian jaw and skeletal features. It lived during the late Triassic period, about 235 to 221.5 million years ago. It was a small, carnivorous or insectivorous cynodont, likely resembling a large rat or a small dog.
What is a common probainognathian?
While many cynodonts show intermediate features between earlier vertebrates and mammals, the probainognathians represent the group that was the last in this transition and the closest to the origin of mammals. Within that group, one of the most classic and well-studied examples of an intermediate cynodont is Probainognathus.
So it is the same as the advanced cynodonts.
The terms are related, but they are not the same thing. Probainognathus is a specific example of a genus within the larger group of advanced cynodonts. The group of advanced cynodonts, known as Probainognathia, includes many different types of species, and Probainognathus is one of the earliest diverging members of that group.
So as a member of the advanced cynodonts it is not an intermediate cynodont.
Your confusion is understandable, as the terms intermediate and advanced are often used somewhat interchangeably in this context. However, they describe different aspects of a single evolutionary concept. A paleontologist would consider Probainognathus to be both an advanced cynodont and an intermediate fossil.
Give an example of a cynipid.
Dimetrodon is a classic and widely known example of a synapsid. Although it is often mistakenly grouped with dinosaurs, it lived millions of years before the first dinosaurs appeared. Dimetrodon was an ancient, sail-backed vertebrate that thrived during the Permian period, roughly 295 to 272 million years.
Give an example of an intermediate between the Dimetrodon and the Probainognathus.
There is not a single intermediate organism between Dimetrodon and Probainognathus, but rather a broad group of advanced synapsids called therapsids. Therapsids evolved from the same lineage of pelycosaurs that Dimetrodon belonged to, but were more direct evidence.
What is an example of an intermediate between the therapsids and the Dimetrodon?
An excellent example of an intermediate between the pelycosaur grade, which includes Dimetrodon, and the therapsids is Biomosaurus. It was one of the most basal and earliest therapsids, and its fossils demonstrate a clear set of features that are more advanced than Dimetrodon but not as fully mammalian as later therapsids like Probainognathus.
Isn't it a therapsid?
You are correct. Biomosaurus is a therapsid. The previous answer was designed to clarify its role as an intermediate between Dimetrodon, a pelycosaur, and later, more advanced therapsids like Probainognathus. In summary, Biomosaurus was a therapsid, but a very early, primitive one, and it represents one of the first branches of the therapsid lineage. In this way, it serves as an excellent intermediate example.
So it is not an intermediate.
You are correct to question this. It can be confusing because Biomosaurus is indeed a therapsid. A paleontologist would consider Biomosaurus to be both a therapsid and an intermediate fossil, and the terms are not mutually exclusive. To be precise, we can say that Biomosaurus was a basal therapsid that serves as an intermediate fossil between the earlier pelycosaurs and the later, more advanced therapsids.
Give an actual intermediate between the therapsids and the Dimetrodon.
There is no single actual intermediate species between the synapsid group that includes Dimetrodon and the therapsids. Instead, the transition is documented by a group of sphenacodontians from which the therapsids evolved. The evolutionary relationship is not a simple linear progression but a branching tree. The split between the lineage that led to Dimetrodon and the one that led to therapsids is thought to have occurred within the sphenacodontians. The therapsids arose from a close relative of Dimetrodon, also within the sphenacodontian group.
What was the close relative?
It's important to clarify that Dimetrodon is a member of the sphenacodontians, the larger group of synapsids that includes both sphenacodon and the direct ancestors of therapsids. Therefore, the relative is actually another sphenacodontian species.
Give an example of an intermediate between Haptodus and Sphenacodontids.
Haptodus is considered to be on the stem leading to both the sphenacodontids, Dimetrodon and its close kin, and the therapsids. Unlike Dimetrodon, Haptodus was much smaller and lacked a sail. This means that Dimetrodon is not a direct ancestor of therapsids. Instead, Dimetrodon and the therapsids share a close common ancestor that was also a sphenacodontian. While the fossil record shows that the transition from a Haptodus-like synapsid to sphenacodontids occurred gradually over time, there is no single species universally recognized as the missing link. Instead, the evolutionary change is represented by a sequence of increasingly specialized basal sphenacodontians. An example of a transitional sphenacodontians is Eunthodon.
Give an example of an intermediate between Eunthodon and Haptodus.
An actual intermediate fossil between Eunthodon and Haptodus is difficult to name because the relationship is not a simple, linear one. The fossil record shows a greater complexity, with both genera found in the same geological deposit, suggesting they coexisted rather than one evolving directly into the other.
The Lanthodon had to evolve into a Haptodus, therefore there must be an intermediate.
That line of thinking reveals a common misconception about evolution, called linear evolution or anagenesis, where one species is thought to simply turn into another. The reality is more like a branching tree, a process called cladogenesis, where a single ancestral population splits into two or more new populations.
So what did they evolve from?
Eunthodon and Haptodus are both basal sphenacodontians, and they evolved from earlier, more primitive members of the synapsid group. The very earliest synapsids branched off from other amniotes, the group containing reptiles, birds, and mammals, during the late Carboniferous period.
What are some intermediates between Haptodus and the ancient ancestor?
Answering this requires looking back to the origin of synapsids, which occurred over 300 million years ago. Haptodus and Eunthodon are both primitive members of the sphenacodontian group, which evolved from an earlier, more basal synapsid lineage. Therefore, the intermediates exist between these basal sphenacodontians and the very first synapsids, whose fossils are rarer. A crucial group of intermediates were the Ophiocodontids. These early synapsids were common during the late Carboniferous and early Permian and are considered to be very close to the evolutionary root of sphenacodontians.
One of the weakest links of the tree model is the fact that random genetic mutations are the only possible explanation. Natural selection only applies within a population where certain individuals do not survive to propagate, thus producing progeny with a given characteristic propagated by the survivors. We know what random genetic mutations produce: disease and defects. We also know that multiple mutations producing a new species or group from another one cannot occur simultaneously because the probability is zero. They must occur over time, therefore, the intermediates must be stable enough to undergo mutation themselves. There are just no examples of these intermediates in the fossil record. Also, the probability that a mutation will produce a stable product as opposed to a diseased, defective one is 50/50ths at best. The probability that a stable intermediate will produce a chain of stable mutated offspring eventually producing a new species or group is nil. The above impossibility is compounded by the fact that there are estimated to be over 20 million species on this earth. the probability that a chain of random genetic mutations producing a new species will occur 20 million times is zero. It is not even reasonable to consider it since, as I mentioned earlier, the only evidence we have regarding random genetic mutation is that it produces diseased and defective products. The only genetic mutation one can consider is akin to gene splicing, where a genetic template is used and variations of it are produced. This is not a random process but rather an engineering or scientific process. Another weak link in the evolution theory is where the rubber finally meets the road, as they say. And that is the case of the single-celled organism, a microorganism or microbe if you like. These organisms have a specific structure and functionality. The simplest description is that of a cell wall, cytoplasm, and a nucleus. Are any of these elements known to exist independently in nature so that they could, in some manner, come together randomly to form the specific structure required? Of course not. It is not even reasonable to consider it as random, for if it were, man would be able to create the simplest of organisms from the raw materials. All man can do is work with the material available, not create it. Creating it requires a technology far more advanced than what we have available. The last weakest link I would like to discuss is some of the physical attributes of life: eyesight, hearing, and mobility. First, eyesight is the result of the manipulation of light rays using principles of physics. Light is passed through a lens and focused onto a receptor. There must be knowledge of optical physics to know that a lens and receptor are required to process light into images and to produce the proper curvature of the lens in addition to the proper medium of the lens to propagate light waves. Can any reasonable person believe a lowly protoplasmic organism knows that certain genetic mutations are required to produce the lens and receptor? Can any reasonable person believe random processes can produce a lens and receptor from a single-celled organism, let alone a complex being housing them? The same is true for hearing, which is the translation of mechanical sound waves into recognizable audio using the eardrum. There is no reasonable argument for random processes producing a complex instrument that uses principles of mechanical physics from a single-cell organism. Nor is there a reasonable explanation for how creatures using mechanical physics via cables, forces, fulcrums, and beams for mobility and other functions can be randomly produced from a single-celled organism. These are a few of the weakest links in the evolution theory. One can do further research into these issues to discover that they cannot be the product of random processes, for the probability of them occurring is zero, especially when the most complex product, man, is at least 20 million species random chains of mutations removed from protoplasm. It is even more impossible when one considers the gap between the various phylogenetic groups rather than species. There is a perfectly simple explanation for why only simple creatures are found further back on the timeline, which I will propose in another letter. When all is considered, even lightly, and when the previous conclusion regarding super advanced technology is remembered, one can only make one decision. The only real question is, how does one decide who or what the creator is?